The assembly of cilia and flagella requires the delivery of large amounts of tubulin to the growing ends of the organelles' microtubules. Tubulin is thought to be translocated to these sites by intraflagellar transport, or IFT particles, protein complexes that shuttle cargo back and forth along the microtubule-based axoneme. But Karl Lechtreck from the University of Georgia in Athens says that tubulin transport and the mechanisms that regulate it have never been studied in detail.
Nobody had really seen high-quality transport of tubulin in vivo. So we expressed the tubulin GFP in Chlamydomonas, where we can very nicely apply turf imaging to see tubulin being transported by IFT, and that worked very well.
Lechtreck and colleagues, led by graduate student Julie Kraft, photobleached the cilia of Chlamydomonas cells so that they could easily observe GFP-alpha tubulin particles moving to the tips of the organelles at speeds commonly associated with intraflagellar transport.
We saw by two-color imaging that tubulin GFP is a cargo of IFT, that it is transported in association with IFT particles from the base to the tip. And we also saw it entering the cilia by diffusion. What we very quickly realized is that there is much more tubulin transported when cilia are growing. So the IFT particles seem to be largely empty when cilia are in steady state or non-growing, but highly loaded with tubulin when they are growing. And that raised for us the question, how is the cell actually regulating this?
Chlamydomonas cells could regulate tubulin loading onto IFT particles at the level of the entire cell, for example by upregulating tubulin expression when they need to extend new cilia. Alternatively, tubulin loading could be independently regulated at each of the cell's two cilia. To address this question, Kraft et al. Generated so-called long-short cells that each exhibit one growing and one non-growing cilium.
Then we image tubulin transport in both cilia, and what we observed is that the cell actually transports tubulin only into the growing cilium. That means it can be not regulated, for example, just by the presence of soluble tubulin in the cell body, but that there must be some additional control mechanism which allows the cell to specifically direct tubulin transport by IFT into the cilium in need. And another implication of that the cell must be actually aware that one of its cilium is shorter and that there must be some kind of signal which ends in higher loading of IFT particles with tubulin cargo.
Kraft et al. Saw the same effect when they manipulated and mated Chlamydomonas gametes to form a tetraciliated zygote with two short and two long cilia. The zygotes equalize the lengths of their cilia by upregulating tubulin loading and intraflagellar transport in the two shorter appendages. Furthermore, Western blotting and fluorescence recovery after photobleaching experiments suggested that upregulating tubulin transport can selectively increase the concentration of soluble tubulin in growing cilia.
Based on our estimates, the concentration of soluble tubulin in the cilium is quite high, above 200 micromolar, which is well above the critical concentration. We think that such a high tubulin concentration, which you could never get by diffusion, is needed because in the cilium you have to polymerize 9 plus 2 microtubules, so 9 doublets and 2 singlets in that very small volume. So you need a lot of tubulin. It will be consumed very rapidly by this axonemal growth. And therefore a constant supply of tubulin is needed to keep cilia growing, and that's what we think IFT is doing, that it kind of functions as a tubulin pump.
This tubulin pump appears to be misregulated in Chlamydomonas mutants that form abnormally long or short cilia. Mutants in the cyclin-dependent kinase LF2 form cilia of variable lengths, including very long ones, and show elevated levels of tubulin transport at steady state. Short flagella 2 mutants, in contrast, prematurely downregulate tubulin transport when their cilia have only grown to a length of 6 microns. So how might intraflagellar tubulin transport be regulated and coupled to cilium length?
Yeah, that is the future question. Currently, we do not know how cells measure the length of their cilia. We know that probably various protein kinases are involved, but we don't know the signaling pathway. So what is sensing ciliary length? What is the mechanism? What kind of signal is then transmitted to the cell to let it know its cilia are of correct length or incorrect length? And how is that signal then translated into differential loading of tubulin onto IFT particles? That is currently terra incognita.
For now, though, you can learn more about how tubulin transport is upregulated during ciliary growth by a cilium autonomous mechanism in the paper by Kraft et al., published in the January 19th, 2015 issue of the Journal of Cell Biology.